Hello, this is Dr. Anna Maria Mihaicha. I'm the president from AM Medical LLC, and today I want to talk to you about Exosomes The New Frontier in Medicine. This part one is going to be about longevity and age reversal. What are exosomes? They are small vesicles that are between 40 and 100 nanometers, so 1,000 times smaller than a regular cell. And they are made from the inward budding of these membranous vesicles. And the mesenchymal stem cell exosomes that we're talking about here are specifically from a perinatal cell. And so these vesicles here is a picture of what we're talking about. So the idea would be, why wouldn't we use stem cells, but we would use the messengers of the stem cells? So one of the things that's been shown is that stem cells are what's called parasites. So they don't actually go and after an infusion of mesenchymal stem cells go and regenerate any specific tissue, but what they do is they glob on to the blood vessels, like is shown here, and they excrete the exosomes with the information that then goes and modifies the healing process in the organism. One of the problems is that the stem cells are removed from the circulation. It's been shown as fast as 12 hours. They get lodged in the lungs, but the majority of them are all gone within 72 hours. There can also be a rejection inflammatory reaction because they are recognized by our body as foreign. They have a cell wall and they do have DNA. Mesenchymal stem cells cannot cross the blood-brain barrier, and so if we want to affect the brain and regenerate people neurologically or rejuvenate them, we need something that's smaller in terms of its ability to go to a site of injury and create a healing process. Also, the stem cells, by virtue of their culture process, are damaged by their culture medium, so they are kept in DMSO, and that is actually toxic to the cells and kills them. Also, the culture medium is kept at 40% oxygen, so it creates oxidative stress. So these mesenchymal stem cells are expressing a senescent or aging phenotype. So it means that we think that we're getting perinatal information, but in fact, due to the processing of the cells, there's aging that already occurs, and with that, the potential for increased inflammation. So what is in these exosomes? We call it the cargo. And there's over 857 unique gene products and exosomes, and greater than 150 kinds of microRNA. And I'm going to go deeper into what this microRNA actually is. So what is interesting in regards to the difference between a perinatal mesenchymal stem cell and the mesenchymal stem cell of an old person is that the cargo is significantly altered. So there's more than 300 growth factors in these perinatal exosomes, whereas there's only half of those growth factors in the adult ones. So exosomes are used by all cells as communication messengers. They're even used by cancer cells in order to promote metastases. They're used by aging cells to modify their neighborhood because they are infecting their neighborhood with age, and that signaling of what's called the senescent secretory phenotype is actually via these exosomes. So inside of the exosomes of the mesenchymal stem cells, we have enormous amount of proteins and information. So we have receptor sites, adhesion molecules. We have very important lipids, phosphatidylserine, phosphatidylcholines. These are very important and have many different biological effects. But then we also have genetic material. So we have messenger RNA, microRNA, and mitochondrial DNA. And so RNA can be infected into a target cell and there create effects. So I'm going to go deeper into that, but the biggest point here is that we can affect mitochondria because we can give youthful mitochondrial RNA and DNA to be able to rejuvenate these mitochondria. And then we can give messenger RNA and make proteins that are youthful. And microRNA it has enormous effect on the host genome transcription. There are other things in there. So aside from growth factors, we have proteins. We have heat shock proteins, especially heat shock protein 70 has been associated with exceptional longevity. There are enzymes in there. So this is a jam-packed little vehicle that acts like a virus in the sense that it is so small that it cannot be seen by our immune system and destroyed. It's not recognized as foreign. And as this tiny little bubble, it can go into a cell and change how that target cell is operating. So microRNAs, about 60% of our human genome is actually regulated by microRNA. So we have DNA, and DNA gets transcribed into RNA, and then RNA is the code to make proteins. But it turns out that the microRNA is really a regulator that determines what is being transcribed and what kind of a protein is being made. So it has regulatory action, and it can either prevent or allow the transcription into a protein. So in many different organisms, it's been shown that microRNA expression changes with lifespan. So these microRNAs appear to also modulate the lifespan, and you can predict longevity by the amounts of certain microRNA that are present in the cells. With age in humans, microRNA expression changes, and it's been shown in these peripheral blood cells that many microRNA levels are declining. So what we're seeing is a loss of regulation of these microRNAs. So for example, some of the microRNAs that are in these exosomes that have to do with the extraordinary effects of neurological rejuvenation. I mean, we are having patients with spinal cord injuries who were quadriplegic, who are able to walk again and have sensation. There is some tremendous work done by Dr. Doug Spiel. He's an interventional pain specialist who infuses exosomes intrathecally, meaning into the spine, and he absolutely has miraculous patient cases where people all of a sudden are able to feel as well as to move their bodies and actually regenerate their ability after having severe spinal cord injuries. So this is an absolute miraculous promise. So the microRNAs are responsible for even neuronal regrowth and remyelination. So what's previously been thought to be impossible to regrow a spinal cord, to regrow brain damage, is no longer impossible. And here are some of these microRNAs associated. 133 specifically associated with increased neuronal plasticity and the neurite remodeling. We have microRNA 21, which is associated with improved survival of these intact neurons. It's neuroprotective in cases of ischemia. So, for example, when people have strokes, there are very well done studies that show that exosomes infusion protect the brain from cell death after something like a stroke. MicroRNA 133b promotes the neuron outgrowth, so the neurons that grow like a tree, and it also improves functional recovery. The microRNA 124 promote productivity of these neurogenic cells, so it makes them more viable, more active. As well as microRNA 12. So these are just a couple examples. So remember I told you earlier that there's more than 150 microRNAs in these exosomes, and so they are able to give information to the target cells and create absolute regeneration. And it appears that the mechanism by which these exosomes are so therapeutically valuable for so many diseases is exactly through this microRNA mechanism. So there are also key immune and growth factors present in these exosomes. There are numerous ones, but BMP5 stimulates bone growth. We have GDF15, it's a growth differentiation factor. So if you're thinking about a growing fetus and these perinatal stem cells, the information that it takes to make an infant grow into a grown-up human is fully functional in these cells. So we have immune stimulating growth factors. We have stem cell factor that is responsible for the stem cell growth. TGF-beta 3 is one of the most important anti-inflammatory proteins. It does convert the inflammatory T cells to the anti-inflammatory regulatory T cells. And why is that so important? Because in anti-aging, it is inflammation that drives the aging process. All chronic diseases, including autoimmune disease, cardiovascular disease, dementia, kidney failure, bone loss, all of those are accelerated through inflammation. So we are able to have profound immune modulatory effects by being able to give TGF-beta 3, there's TGF-beta 1 and others as well in exosomes. VEGFs is vascular endothelial growth factors, so you have the support to grow new blood vessels. The body and the tissues need oxygen delivery as well as nutrient delivery, and then we also have other binding molecules like ICAM and others. The list is extraordinary. In one of my brochures I have the extraordinary list of growth factors that are present in our exosomes. It is quite impressive, all of the targets that can be affected. So the therapeutic effects of these exosomes, they can influence the growth of a target cell. So remember, I told you that this little exosome can go into a cell and then infect it with its information of youth. So it influences the phenotype. What does that mean? Your phenotype is what you're epigenetically expressing right now. So for example, if you're an old man or an old woman with a lot of medical problems, then that is your phenotype. So we are modulating that expression of age, and we are artificially reversing it to youth. So your cells are now under the control of the information of perinatal microRNA exosomes. The exosomes can also remove cytotoxins from the cells. So we're talking unwanted proteins and cytoplasmic DNA. What good does that do? So as you know, Alzheimer's dementia, for example, has unwanted proteins in the cells, the tau protein. So they have been shown to be able to be removed by these exosomes. Hence, you have improvement of cognition in chronic neurodegenerative diseases like dementia. They contribute to the cell fate decision. So cell fate would be, does this cell, is it locked in senescence? Is it locked in inflammation and aging? Is it about to die? So by giving it these microRNA, we can change that regulation and tell the cell to rejuvenate itself, as well as to improve its overall function and longevity. The exosomes do promote regeneration, profound. We talked about neuronal regeneration, but this is applicable to all tissue types and all organs. Particularly in part 2 of this lecture series about the exosomes, I will be talking about regenerative capacity for all chronic diseases and all organ tissues. There's a profound immune modulation effect. So we discussed earlier that because of the specific transmitters and messengers that are there, there's an anti-inflammatory effect that occurs. So that makes these exosomes extremely useful in things like autoimmune disease, chronic Lyme disease, diseases that otherwise have been thought to be very difficult to control. There are some extremely promising findings with the exosomes. Anti-inflammatory properties are important for all kinds of things. So we have osteoarthritis, rheumatoid arthritis, all of the pro-inflammatory things like chronic bowel diseases. Anti-fibrosis. Fibrosis means scarring. For example, chronic kidney disease is progressing through scarring of the kidney tissue. So we are halting that aging process of organ scarring. So what tissue effects do the exosomes have of these mesenchymal stem cells? So they've been shown to be protective on the heart. They've given in studies artificial heart attacks to mice and then injected exosomes and proved that these exosomes protected the heart muscle from dying and had significant effects to not give these heart failure progression symptoms. We have studies with cartilage regeneration, repair of lung injury, reduction of kidney fibrosis, studies of reversal of type 2 diabetes because the insulin resistance is downregulated and the destruction of the beta cells in the pancreas is reduced. We can reverse the degeneration of neurons, astrocytes, and then the synaptic loss in the hippocampus. The hippocampus is in charge of our memory, also has all the stem cells of our brain, has a large storage of those in there. The axonal growth of these neurons in our brain can be enhanced. Our neurological function can be improved, and there's much more that I'll go into in part 2 as well. The studies that have looked at the target tissues that exosomes could affect is the heart, the brain, the liver has significant benefit, the lung, the intestine, skin and wound healing. There are extraordinary results of regeneration. If you understand that TGF gene is the gene that the salamander uses to regenerate its limbs when you cut it off, and this is what we are transfusing in these exosomes. Limb ischemia, so peripheral artery disease, arteriosclerosis, there are benefits there, skeletal muscle regrowth, spinal cord injury, and immune system dysfunction all have benefits. The advantages of the exosomes as compared to stem cells, so they can be transfused intravenously. We give them as a slow IV push. The whole procedure takes about 15 minutes. They can also be given intranasally, so it's been shown that exosome can go through the cerebriform plate, which is above the nasal septum, and can go directly into the brain. They are unique in that they do cross the blood-brain barrier, so they're small enough that the information can also go into the brain. So they deliver microRNA and messenger RNA. They can do something that's called homing. What is interesting is that these exosomes have been shown to be intelligent. They know exactly where the injury site is and go there to repair. For example, there were studies where functional MRI scans were done after infusions of stem cells compared to infusions of exosomes. The stem cells didn't even make it to the brain at the area where the stroke was, but the exosomes showed up on the functional MRI scan. So they're very, very intelligent. They are not perceived as foreign, so there's no rejection reaction. And there is no removal of the exosomes in the lung as occurs with the mesenchymal stem cells. They do not transdifferentiate. So one of the problems with particularly embryonal stem cells is the concern for malignant transformation. So since we are not infusing a cell and we are not infusing DNA, there is no risk for cancerous transformation because what we're infusing cannot go down that path because it is not a cell. And the potency of the exosomes is really related to its useful functionality. So we don't want to infuse exosomes from an old person, but we're using perinatal, absolutely functional mesenchymal stem cell exosomes, which have the highest functional capacity. So let's discuss a little bit. So the hallmarks of aging, there are 9 hallmarks that have been shown to be related to aging. It's never just one particular pathway. Anyone who says, I found the one immortality gene and the one way that this works, that's incorrect. So genomic instability has been related to aging. Telomere shortening. Our epigenetic are altered through, for example, environmental toxins like lead, cadmium, arsenic. Our environment, our choices are epigenetically significantly changing our expression. In fact, our genes, in that sense, the inherited information only plays a very small part compared to the environmental epigenetic effects on our genome. Loss of proteostasis is the idea of functional proteins in our body that when we start making proteins that don't fold well or have problems, we cause disease, for example, in Alzheimer's dementia. Deregulated nutrient sensing means, for example, insulin resistance. It's one of the prime factors that lead to the development of diabetes mellitus. That is an important aspect. Mitochondrial dysfunction. Mitochondria are the powerhouse of the cell. If we cannot adequately make energy to give optimum energy to all cellular function, then we have a progressive degradation. Senescence is a word that means, in fact, aging. It is related to the fact that cells progress in this aging pathway and at some point reach a point of no return. That they start secreting all of these inflammatory cytokines that infect all of their surrounding with the information of aging them as well. Stem cell exhaustion. Our stem cells, as they're aging, they become dysfunctional, and that is why we want to use perinatal stem cells, exosomes, to rejuvenate our body. And the altered intracellular communication, so that's related to these microRNAs that I talked about earlier. It's been clearly shown that in the peripheral blood of humans that are older, those levels are reduced. So let's look at this study in which the effects of young exosomes that were injected in aged mice. And what they did was they used exosomes from mice that were 12 to 18 months, and they used exosomes of 3-month-old mice. So first they looked at each of these exosomes from the young and the old mouse and what microRNA was it expressing. And comparatively speaking, there are several microRNAs that were reduced in the old mouse. And it was reduced in specific tissues like in the lungs and in the liver. So then what happened was that the young exosomes were injected into the old mice, and they measured the microRNA levels, and it turned out that they were reversed in the lungs and the liver. And what they also wanted to know is, did the genetic markers of aging change at all? So P16, mTOR, IGF, were downregulated. And in the next slide, I'm going to explain that. So the conclusion is that the exosomes from the young mice could reverse the expression pattern of the aging-associated molecules in the aged mice. So these markers of aging, the 4-week-old mouse was injected into an 18-month-old mouse. And these are the markers that were looked at. So P16 is a regulator of the cell cycle, and it's a marker of senescence. Remember, senescence is an aging process. The mTOR gene is a regulator of aging and longevity. I've talked about that quite a bit in my immortality video. It's a target of rapamycin, and we want to downregulate it because if it is upregulated, then all of the diseases of aging start happening, like arthritis, diabetes, heart disease, cancer. So in this case, in this study, it's been shown that the mTOR expression was down-regulated, which goes along with reversing aging and prolonging lifespan. Insulin growth factor, the reduced expression is also associated with longevity, so it was suppressed in this study. And then the other thing that they looked at was telomerase-related gene expression. So telomerase is the enzyme that is in charge of repairing the telomeres, which are the little hairs, the capping structures on our DNA. And the more telomerase you have, the greater your ability to repair the telomeres. So that's associated with prolonged survival and healthy longevity. So all of these markers were actually affected towards longevity and youthfulness in the old mice. So we are applying this very same concept. What was also interesting is these are the mice, and you can see that these exosomes that were marked with a fluorescent stain, that they went everywhere. They went into every cell and took care of business there and started working. You can see in the organs, in the liver, in the lung, in the brain, they were able to go everywhere. So one of the newest articles that just came out was found that in the exosomes there is an enzyme, NAMPT, that's been looked at because it declines in mice and in humans when people are aging. And so it promotes NAD, which is major molecule of the energy metabolism in our cell. And so increasing this enzyme counteracts aging and extends healthy lifespan. And this is contained exclusively in the exosomes in mice and humans. And so giving exosomes can increase the levels of this enzyme. And what happened was that these mice of the aged mice, they started to have better physical activity. They had multiple cognitive abilities that were increased. So this is a avenue of anti-aging intervention in humans, and this is brand new from August 2019, Cell Metabolism. This was published. So the idea is that these enzymes in the exosomes are affecting the pancreas. So you have the biosynthesis of NAD, nicotinamide, increased. In the retina, so these mice had better vision and photoreceptor function. In the hippocampus, you had improved cognitive function, better learning, you had increased lifespan, decreased aging, and you're also modifying the SIRT1 gene, which is a major gene in regards to longevity and aging that regulates inflammation. Sleep quality improved and physical activity did improve. So we've discussed that these exosomes, in terms of therapeutic interventions, we have a donor cell, which is our exosome producing mesenchymal stem cells, so it's perinatal. These vesicles contain all these microRNAs. They infect the new cells and cause messenger RNA translation into new proteins. MicroRNA can either inhibit certain regulatory proteins and we are now transcribing proteins that are healthy and that are fully functional due to the information given to us by the perinatal stem cell exosome. So these anti-aging and longevity affects all of the different organs. So it affects the phenotype, the expression. They transfer the molecular payload and translate this messenger RNA. Then we also have a regulation that post-transcription, so when we transcribe our DNA, there's a regulation of this microRNA on how we're doing that. And in fact, it's been shown that we're now able to transcribe our own DNA much better and cleaner. And there is a direct protein action within the recipient cell from the exosomes. So, in conclusion, the aging results in a loss of functional capacity in a number of key organs. The microvesicles that are derived from stem cells can become a novel cell-free therapeutic intervention that can mitigate the undesirable effects of aging by enhancing tissue regeneration and improving the human health span. So we have here a possibility with extraordinary regenerative and age-reversing properties. This is the absolute new frontier, and the effects that we are seeing are extraordinary. I had my exosome infusion myself about a week ago, and it's been a quite fantastic journey. And I think that to know now biologically how to modulate our own aging cells in so many effective way, the idea of longevity and immortality combined with mindful progress into the future is definitely at hand. So MSC exosomes can affect all hallmarks of aging. They have profound rejuvenating effects, and this is the broad therapeutic effects that occur due to this horizontal transfer of RNAs and proteins. Thank you very much for watching today's lectures. I hope you enjoyed this information about our new treatment modality at AM Medical, the exosomes. Stay tuned for a part 2 in which I will be discussing the ability to treat all chronic diseases through exosomes. Please follow us on Facebook. Check out our website at ammedicalmd.com and subscribe to our YouTube channel, AM Medical LLC. Thanks again for watching. Bye-bye.